Thank you very much to Francisco, John, Brian for inviting me to the symposium. The subject's really interesting and close to my heart. In the early 1970s, when I started to work on cultural evolution with Luca Cavalli-Sforza, the reason was the furor that was happening at Stanford and Berkeley in connection with Arthur Jensen and Shockley about the very high heritability of intelligence and its so-called consequences for education. So our first study, which was published in 1973, actually showed that if you used the classical twin MZ, monozygous, dizygous twin correlations obtained under assumptions of purely cultural transmission, non-genetic transmission, that is, gave you the same correlation estimated from heritability that you could get from purely genetic models. There is a modern update of that, which is currently in press in a strange place in the Israel Journal of Ecology and Evolution, because they have a special issue on cultural evolution. And it goes into great depth into why those twin correlations should lead to an estimate of heritability when it's only cultural transmission from parent to offspring. Now, it's not everybody who thinks that cultural transmission is important. In fact, when you look at one of the most important books ever written, Darwin said, "I have not met with any evidence in support of the transmission of superstitious customs or senseless habits." We all know that that's not true, and that much of the senseless habits and superstitions are actually culturally transmitted, either vertically or horizontally or obliquely. And on the other hand, if we ask what is the process of natural selection, there is a considerable debate, particularly among the population geneticists, about the way in which natural selection occurs. And Burian, who is a leading philosopher of evolution, claims that natural selection is the process of solving problems posed by the environment. On the other hand, Lewontin, in a very influential paper in 1983, introduced the notion of construction, that organisms construct their environments. And that paper, although it was not quantitative in nature, was the reason that my colleagues and I started to work on niche construction in the early 90s. And we came up with a mathematical theory for niche construction, which occurs when an organism modifies its relationship with the environment, and that modification can be passed on to its offspring or affect other species. That niche construction, if some genes provide material which alters the way in which natural selection acts on other genes, then the dynamics of natural selection change radically, especially if those effects last over time. And that was the subject of our book. On the other hand, there hasn't been much done on how some cultural traits construct a niche in which other cultural traits, or for example, genes, could evolve. And that was the subject, and I think this is the first paper on this subject that we wrote with my postdoc, Yasuo Ihara, who's now on the faculty at University of Tokyo, on exactly that, how cultural transmission of one trait could depend on what was happening at another trait and therefore influence the evolution. So one would have a kind of epistasis in culture. That's a term invented by R.E. Fisher, although he called it epistasy, which is the interaction between different genetic traits in his terminology. But in culture, it could be just as interactive. So let me give you an example of the kind of thing that can happen. And this was a study we did with Melissa Brown. We've been studying the Japanese records from Taiwan. During the occupation of Taiwan from 1895 to 1945, the Japanese kept absolutely meticulous demographic records of every person in Taiwan. What we know is that in the colonial era, there were ethnic groups in Taiwan that were either Han, they had come starting in the 1600s, or indigenous people, Aborigines, who are actually Melanesian in origin, as genetics shows. But the mainlanders are not involved. That is the people who came after the revolution. And those ethnic groups lived side by side with very little intermarriage for a long time. One of the reasons why was the Han bound the feet of their women. These are some of the most dreadful photos you can see about cruelty, and the different ways in which the women's feet among the Han were bound. What happened is that the Han regarded the indigenous people as barbarians because they didn't bind their feet, and they would not choose to marry them because of that. Now, why did something like a change in foot binding have dramatic consequences for a lot of other cultural phenomena that happened shortly afterwards? In 1915, the Japanese outlawed foot binding, and they did it because they needed the women to work in the fields and the men to work in construction and on the railways and things that they were doing to develop the food supply for them, for Japan. So what happened is really quite interesting, that prior to that, among the indigenous people, there was quite a high fraction of what we call uxorial local marriage. Uxorial local marriage is where when the couple marry, the boy goes to live with the girl's family. Very rare in rural China and very rare among the Han in general. It goes in modern rural China, it's about 5% to 3% of such cases. Almost every case, the girl goes to live with the boy's family. So after foot binding, what we see is this radical drop until 1945 among the population of Aborigines in who they're marrying and what their living arrangements are going to be after marriage. This is the proportion of uxorial local marriage in the Aborigines, and this is the proportion in the Han here. Radically different. Foot binding had a very strange consequence. It changed the way in which marriage patterns occurred. But that's not all. It changed religious practices to the extent that now that women had to work, the medicine people, the shaman, who in the Aboriginal community had always been a woman, suddenly could not be a woman because there would be nobody to look after her children if she was working as a shaman. So within a very short time, the shaman became a man. And in the interviews that Melissa did in the 1980s and 1990s, the women still believed that the shaman should be a woman, but it wasn't the case because they couldn't, because of this phenomenon of who looks after children. So this is an example of a trait, foot binding, which apparently shouldn't have much to do with marriage or with who should be the shaman, causing these phenomena that later happened. So that foot binding was a niche, it was a trait that actually had a constructive effect on these other traits. Now, how could you go about trying to formalize this? And this is why I call it next generation. We've been for 40 years since we started the theory, been working on relatively simple models of cultural transmission. Here is a three-trait model where we have one trait, which is this T here, which is a focal cultural trait, another cultural trait which determines the selection that acts on that trait, and a third trait which determines the level of homophily on that trait. Now, homophily in our terminology is assortative mating. And in connection with what you just saw from James Fowler, we proved in 1985 that you can have complete homophily, that is self-fertilization in plants, And that will allow for up to 50% selection against the trait that you're homophilizing, and the trait will still advance. It will still succeed. And that's because of this fight between the process of assorting and the process of selection. It's a general phenomenon. It happens when you do it genetically or when you do it culturally. Anyway, these three traits are what we're going to focus on. Homophily here is the level of assortative mating. And it's complicated because we now have three traits that are going to be culturally transmitted. All of them are culturally transmitted. These are the rates of transmission. Unfortunately, when you switch from one culture like Apple to another culture like Microsoft, you end up with this kind of thing here. What you see here are the three traits, their rates of cultural transmission, the relative fitnesses of the trait big T, determined by its structure at the second position, the second trait, and the level of assortative mating will be determined by the third trait. So we have three cultural traits. You can think of it as a three-locus problem, except nothing is genetic. The homophily does this. What it does is it causes these individuals to choose to mate with individuals who have the same T. And it's not the general form of homophily, which could be done under horizontal transmission, where you choose to contact individuals that are the same as you. But the general principle will be the same if it's done by virtue of mating. So, what's the dynamics under this wild system? It's a very complicated problem. This is one example, and I've given you the parameters up here of this system. And to my knowledge, there aren't very many evolutionary systems in genetics. There are very few where you end up with cycles. And this is one example where we end up with this strange cycle. And you can see that only certain of the genotypes, of the culture types are present. What happened to the, I can't, luckily. So, the green one here, the red one here, and these two dashed, red and green, that one and this one. So only 4 of the 8 phenotypes are present on that cycle. The rest have disappeared under the evolution. Here is a very peculiar case, which I don't think has been seen before, where under this parameter set here, you have either a cycle, depending on where you start from, or you go to this fixation state. So capital T, capital S, capital M, which is this one here, has a domain of attraction, which is specified by the red arrows, and the blue arrows specify a domain of attraction of an orbit, a cycle, which is really quite interesting. So it depends on the starting conditions in the cultural domain where you end up. Sometimes you get a polymorphism with all of the phenotypes there, but interestingly, you don't only go to that polymorphism. So here is the polymorphism, that red square, sorry, the pink square, but at some points, from some starting conditions, you also go to fixation of one type. So here we have history-dependent dynamics. Depending on where you start from, you either maintain cultural variation or you lose it all. Sometimes both can happen. So this is an example for these parameters. The rest of the parameters are specified here, but if you vary the parameter C, for instance, in this range of the C's, you get a polymorphism. In this range here, you get that kind of oscillation. And up here, you get fixation of this kind here. This is another way to represent the different kinds of dynamics that can occur under this very complicated model of cultural transmission. This is an example of the cycle of all 8 that was in that dark gray area. This is an example of the same thing, only this is a special case where we assign the assorting and selection to be determined by the same cultural trait. So this is only 4 dimensions. And what we wanted to do here was to compare the case where we had purely genetic determination of the culture with what happened when you didn't have genetic determination of the culture. And the dynamics are really quite different. In this particular example here, for instance, you get a full polymorphism, and over here you get no polymorphisms. But that's only under genetics. So genetic forces a restriction. And in no case where you have genetics determining the cultural transmission have we found a polymorphism. They only come when you have non-genetic transmission. So it's like a razor's edge. Mendel's rules have been shown before to be incredibly special in the world of transmission parameters. And this is an example to show how sensitive transmission parameters are. I just picked this example out of a host of examples that I have in my drawer. You have a trait with three kinds, and I call them seams because I don't like memes. Memes gives some impression of biology, which I do not want to do, and seams come from semiotics. So they're the symbols. And what we do here is we have 3 seams. We have the mating frequencies, which are random mating, and then we have the offspring probabilities of getting these types. Now, all I do is change these by 5%, at most 5%, and the dynamics produces an equilibrium over here that looks fully genetic. In fact, that was what Wright called Castle's law, where you had equal gene frequencies and you ended up with a quarter, a half, a quarter. And over here, we get something that doesn't look at all genetic. And all we did was change these parameters by a very, very small amount. Now, one of the things that has been controversial in anthropology for a long time is whether active teaching occurs in hunter-gatherer populations. Now, next month, it's already online, is a whole issue of theoretical population biology devoted to the evolution of learning that's edited by Kenichi Aoki of Tokyo and me. And that has a number of different approaches in that volume, a number of different approaches to how one can model whether learning should be advantageous in fluctuating environments, in spatially varying environments, etc. But there's been very little done on the evolution of teaching. And one of the reasons for that has been the belief that it doesn't occur much in hunter-gatherer populations. Now, one of the very few people who's looked at teaching in hunter-gatherer populations is Barry Hewlett. And he has some beautiful videos which he's collected over the last 30 years of his field work, unfortunately, in Central African Republic, which is not a great place to do field work anymore, on the pygmies. And he's undoubtedly one of the best authorities on behavior, human behavior in the pygmies. And in that study, this is an abstract of that, he describes vertical transmission being important up to a certain age, and in his movies you actively teach, the adults are actively teaching, and older children are actively teaching younger children. And then after that, it appears that horizontal transmission takes over. So how would we go about asking the question, how does teaching evolve culturally? So the first model we did, this is a study which was submitted last week. The first model, we call it altruistic teaching, and the reason we call it altruistic is because it costs the transmitter to transmit. So that's the meaning of altruism in this context. Joe Wakano is a former postdoc of mine and student of Ken's who's participated in this project. And the first model is asexual reproduction, uniparental transmission. And the very simple notion, actually, that one of the traits we're interested in is a teaching gene, which has alleles big T and little t. And then what is transmitted is a bar or a not bar phenotype, and teaching transmits at rate alpha. Non-teaching, you transmit at a lower rate beta, but there's a cost to teaching as an adult. But the trait that you're transmitting is a good trait. It gives you a fitness advantage. Now, we also have another parameter, which is the cost to offspring development. The development of the neural substrate in the offspring to make it susceptible to teaching is introduced by this parameter here. So we now have 4 phenogenotypes. The T with a bar on it, the T without a bar, the little t with a bar, and T without a bar. So we can study the dynamics of these and ask, should teaching evolve? Teaching being this gene here, and under what conditions would it evolve? Or, well, let me go on and tell you that in that asexual model, the dynamics is rather simple. It turns out to be a linear fractional transformation, which you can iterate completely, so you don't have to do any computer work here. And we have fixed points, which are either absence of teaching and absence of bar, or absence of teaching, or absence of not teaching. And I've told these colors here because you're going to see some different dynamics as a result of these assumptions. But I'm going to compare them with the case where the rule of transmission involves both parents. Instead of uniparental transmission, we'll have biparental transmission with sexual reproduction. And that involves this very simple model of sexual reproduction, that these are the parents, these are the transmission rates. Notice that big A can't transmit a little a because it's genetic. This could be written as T, big T and little t. These are things in our first draft. We didn't use T, we used A. So just in your mind, think of that as big T, little t, little t. So these are the transmission parameters that you need to introduce. Now this analysis is too complicated with the general transmission parameters, so we just assume that alpha D, which comes out of a mating in which both parents have the bar, is twice what it is when only one parent has a bar. So what I'm going to show you has that assumption, and the same for the beta parameter. So up here we see the asexual case, and down here we see the sexual case for one set of parameters. In that set of parameters, you don't see much difference, except here. And in that particular place, you have both the green and the purple. And if we go back and we see what green and purple is, it means the only teachers where there's a polymorphism of the bar or no polymorphism of anybody being simultaneously stable. So this is a bistable situation. Similar to what we've seen from Dr. Binmore before. There are 2 stable fixed points. Now here is a very interesting case, a different case, where you can have what you saw before, but also the polymorphism. The white is where you have all of the traits there present. That can be extended so that now we have both the teaching and the non-teaching stable with the bar, not bar. There are 2 fixed points on the boundaries of the simplex. We have polymorphism for that parameter set, depending on where you start from. Very complex dynamics with a relatively simple system. And this one, the polymorphic area, gets much bigger with this set. These are all the starting conditions under which the alpha and the beta will give you the polymorphism. Really a very interesting kind of dynamics. Now I want to tell you about the mathematics just in case you're interested in. In the green and blue and purple areas, we can completely do the mathematics and show you what's the size of the green and the blue and the purple areas. We cannot find the white area analytically. So that's the fact that it exists is a result of doing the computer simulations which iterate the transformations. That's for honest disclosure. There's another one where the area of polymorphism is much, much bigger than it is in the other situations. I'm going to conclude with a different model altogether, and this doesn't involve natural selection. And this is the first time, to my knowledge, that there's been an analogy between what goes on in drift models with the Wright-Fisher model and the Moran model, some of which you heard in the talk on cancer. And this is the first time that it's been applied to different models of cultural transmission, asking how fast do the traits evolve and how fast do they accumulate. And one of the reasons we ask this is because one of the most commonly cited differences between culture in animals and culture in humans is the accumulation of culture in humans, that it doesn't happen in animals. So that was one of the objectives. I'll just show you about rates here and something about, at the end, about accumulation or variation. So this is from Lycett and Gallett's famous paper in archaeology about different modes of cultural transmission, and they cited our book on cultural transmission. But the interesting thing about this is that they're talking about what happens to the rate of change when you have acceptance of innovation, variation between individuals, etc. So under these conditions here, if the transmitter is the parents and the receiver is the offspring, and you have moderate acceptance of an innovation, then you'll have high variation between individuals, high variation between groups, and slow rates of cultural evolution. Now that's actually taken from a paper we did in 1973. Interestingly, this one up here is where there is a single teacher in the population at any point in time transmitting to everyone. So we called that, sorry, that's the reverse. This is one-to-many, the teacher in the population transmitting to everybody. This is where everybody transmits to one person. Everybody transmits to every single person. So this is a case where you might have conformism, for instance, as we described it in our first paper. So, how are we going to handle this? And this is one of the ways in which it's next generation cultural transmission. We're not going to deal with simple 1, 2, or 3 traits. We're going to deal with many, many cultural traits and ask how does that fit with the most widely used genetic model for evolution, the infinitely many sites model. So maybe one of his most famous papers, Kimura wrote this stuff about the genetics in 1969 of the infinitely many sites model, which he later did a lot of work on with Tomoka Oto. And it's generally accepted as one of the key models for asking questions about whether selection occurs, what's the site frequency spectrum for the frequencies of the different SNPs, single nucleotides in the population. So in the analytical model, there are an infinite number of sites at which mutation can occur. Mutations occur only at monomorphic sites. They only occur in newborns, and the traits have no selection. They're neutral. We take finite sites because we find it rather difficult to simulate infinitely many of them. Mutations will occur only at monomorphic sites. Innovation is happening instead of mutation, and they're neutral. There's no selection here. And the parameters are much fewer than we had before. In fact, I'm not going to even mention this social learning fidelity. That's a separate section in the paper. But population size, the number of acquaintances or the number of individuals you're going to model yourself on, and the maximum possible number of cultural traits, which we now call the imagination space. And for most of the work we do, it's either 170 for random reasons or 200. So we imagine a vector of traits, presence, absence, 200 long. That's our cultural vector. And how would you go about doing this? Well, we do a cultural Moran model. And that consists of the following. At each time step, one individual is born. You saw some of this in this morning's talk. The newborn acquires a variant of each cultural trait from the older individuals by social learning. And then one random individual from the older population dies. So the population goes back to being the fixed size that it was. The life expectancy of a newborn is always n time steps. Now, the Moran model differs a bit from the Wright-Fisher model in some of the properties of the underlying Markov chain. The leading eigenvalue is a bit different, and the leading eigenvector is a little bit different. But in general, if you substitute n time steps in the Moran model, you end up with something that's very similar to the Wright-Fisher multinomial sampling model. So here is the rate, R is the reciprocal of the average interval between fixations. That's the rate of evolution. So here is a picture of how it happens. So this individual has a mutation which occurs at rate 1 over n in our population. It arises when it arises, it's in one individual, so its frequency is 1 over n. And that one goes by accident up to there. All the others here are being lost, which of course happens with probability 1 minus 1 over n. And this one shot up there. So this is the average of this rate between those traits is what we're actually after for the rate of evolution. So let's do it more simply. This is Kimura's formula, actually. So here is 3 fixations. Total time, that one took T1, that took T2, and that took T3. And so this is the rate, 1 over this formula here. So that's what we're after in all of these simulations. So let's take an example where the number of cultural traits is 4. This is a 4 vector, and there's going to be innovation, which could occur in every individual, but only in monomorphic sites. We call it a constrained model because we haven't yet had time to change this into a more sensible term. That has back mutation. So here are 5 individuals in the population, and they all look different. So the transmission modes are following. Here's random oblique learning, best of K learning. You'll see exactly what these mean. Success biased learning and one-to-many. I won't have time to get to one-to-many today. So here is a newborn. All newborns are blank. They don't have anything. They're going to copy a random individual from the population and then replace that individual over here. That individual dies. And then this is the Moran model. Success bias, copy the individual with the most ones, assuming that 1 is a good thing. So here is a group that you've chosen of size 3 that you're going to base your learning on. And in that particular case, you chose the ones from here and from here and from here. That's a success-based model, and you do exactly the same thing as replacing a new individual that you pull out and kill. The best of K models is a different model altogether. In that one, you copy the ones from all positions with ones after you've chosen the K that you want to look at. So this particular, here are 3 individuals you've chosen, and you copy all the ones in that group of individuals. Now in the best of K, we can actually write down the analytics fully. And this is the Markov chain, and this is the solution to the Markov chain, actually, that gives you the complete dynamics. And we can ask, how does it affect the cultural rate depend on K, the number of individuals you decide to copy? Big difference between 1 and 2. If you copy 2 individuals, you get this very big rate. This is the random oblique model. This is the k equal 2, best of 2. And yet not so much difference between 2 and 3. We don't know why that is. There's still a bit of a mystery. And if you want to make this more general, and for instance have one teacher and non-teachers in the population, then to make an analog to Kimura's rate of evolution, you have to take account of the mutations that occur in the teacher and in the non-teacher, and then make a weighted average of those to get the rate. So there is a little bit of work to do here. So we simulated this problem because we know the answer with infinite states. We have exact answers from all of Kimura's theory and our own. We know exactly what the probability of fixation is, the expected time to fixation, the mean conditional time to fixation, given that you haven't fixed. All of that can be solved analytically. But nobody has solved the problem with finite number of states. So that's why we did the simulation and want to ask, how do they compare the infinite states model from Kimura and the finite states model? So we chose, in this particular case, m equals 170. We did an agent-based simulation. So we're actually using a population of 500 individuals, and we've done it for up to 2,000 individuals, where everything that I've shown you is done step by step using each of the individuals as one of the agents in the simulation. So this is the result of the rate of cultural evolution, and this one down here is the simulation result for one-to-many transmission. This is the simulation result for random oblique, and this is the simulation for the best of k, in this particular case, where k was 3. Here are the simulation results. Those were the simulation results. These are the analytic results that we got with an infinite number of sites. There's no difference. This one has never been done with an infinite number of sites. It's too hard to do. But the bottom line here is that the agent-based simulation with 170 sites gave you exactly the same, to our degrees of approximation that are legitimate, the same results as with an infinitely many number of sites. I'd say that's good agreement. Now, Watterson in 1975, he was my master's advisor, by the way, in Australia, wrote a magnificent paper, a paper that is one of the most important in population genetics theory. In that one, he proved, he derived the distribution for the number of segregating sites under the infinitely many sites model, and he computed the mean and the variance in the number of segregating sites. And this black line here is Watterson's distribution. The pink is our 500 samples, sampled from that distribution, from Watterson's distribution. And the blue is the results from 500 samples with a vector of size 200. Up here is the fraction with this many segregating sites. So where they overlap is brown here. So the distribution is not exactly the same, but if you look at the mean and the variance, they're incredibly similar. So this was really unexpected for me, that the distribution from the segregating sites model under the infinitely many sites model should look so similar with 200 traits, so that it's effectively 200 is the same as infinitely many as far as the mean and the variance is concerned. That gives us very good reason to assume that we can do simulations of very large vectored cultural traits and get, with large enough population size, of course, that you can get results which are similar to what you would get under an infinitely many sites model where the problem's been solved completely. And these are my collaborators. Nicole and Laurel are postdocs with me at Stanford. And Ken and Joe are now moved to Meiji University in Tokyo. Thank you.